Hello, welcome to BioTanime. Immune tolerance is a state of unresponsiveness specific for a given antigen, which is induced by prior exposure to that antigen. Tolerance is divided into two: central and peripheral. Central tolerance will limit the development of autoreactive T and B cells, induced by antigen in generative lymphoid organs and high affinity recognition of the antigens. Immature lymphocytes undergo apoptosis upon encounter with antigens, or it eliminates high-affinity self-reactive lymphocytes. Some self-reactive T cells that encounter self-antigens in the thymus develop into regulatory T cells, and immature B cells in the bone marrow change their receptors. Peripheral tolerance causes deletion, energy or regulation of lymphocytes that can recognize self-antigens. Mechanisms of tolerance induction. 1. Clonal deletion. Physical elimination of cells from the repertoire during their lifespan. Functionally immature cells of a clone encountering antigen undergo a programmed cell death. Likewise, differentiating early B cells become tolerant when they encounter cell-associated or soluble antigen. 2. Clonal energy, down-regulating the intrinsic mechanism of the immune response, such as lack of co-stimulatory molecules or insufficient second signal for cell activation. Recognition of such antigens may lead to signaling block and engagement of inhibitory receptors. Also, B cells, when exposed to large amounts of soluble antigen, down-regulate their surface IgM and become anergic. These cells also upregulate the FAS molecules on their surface. An interaction of these B cells with FAS ligand-bearing cells results in the death via apoptosis. 3. Suppression, inhibition of cellular activation by interaction with other cells. Example: regulatory T cells. Regulatory T cells are CD4+ cells that express high levels of CD25 or IL-2 receptor chain. Generation requires a transcription factor called FOXP3. General properties of tolerance. 1. Immature or developing lymphocytes is more susceptible to tolerance induction than mature one. 2. Tolerance to antigens is induced even in mature lymphocytes under special conditions. 3. Tolerance of T lymphocytes is a particularly effective for maintaining long-lived unresponsiveness to self-antigens. Types of tolerance include ignorance. It can be shown that there are T cells and B cells specific for autoantigens present in circulation. These cells are quite capable of making a response but are unaware of the presence of their autoantigen. Or the antigen may simply be present in too low concentration. Since all lymphocytes have a threshold for receptor occupancy which is required to trigger a response, then very low concentrations of antigen will not be sensed. Immunologically privileged sites. Sites in the body where foreign antigens or tissue grafts do not elicit immune responses. Examples are brain, eye, testis, uterus or fetus. Tolerance and transplantation. Tolerance to tissue and cell antigens can be induced by injection of hematopoietic stem cells in neonatal or severely immunocompromised animals. Also, grafting of allogeneic bone marrow or thymus in early life results in tolerance to the donor-type cells and tissues. Such animals are known as chimeras. Thank you for watching the video. For more videos, please subscribe to this channel and press bell icon. See you again in the next video.